Good morning. It's 9 a.m. here in San Diego, so we will begin the webinar. Thank you very much for joining us. Today's webinar is on fragment-based drug design, where we'll cover fragment docking, fragment screening, and then in a scenario where you may have a lead with a crystal structure or NMR structure, where you can start growing your fragment into a drug, and also linking one or two or more fragments in a binding pocket. My name is Andrew Ori, and I'm Senior Scientist here at MoleSoft. If you have any questions after the webinar, please feel free to email or call. During the webinar, you can use the question panel, or you can raise your hand and I can open your mic. You may have already downloaded this software. This is the last webinar in our series. If you've downloaded it, you need to send me the host ID, and I can send you a license key, and it will run on Windows, Mac, and Linux. The webinar recordings are available on our YouTube channel. If you just go to YouTube and search for MoleSoft, you'll find the recordings, and we'll upload this one later on today, probably. Just a quick slide about MoleSoft itself. MoleSoft was founded in 1994 by Ruben Abagayan, who wrote the first code for ICM. And at MoleSoft, Max Totroff is our principal scientist, and Eugene Rausch is our principal developer here at MoleSoft. If you're wondering what ICM stands for, it stands for Internal Coordinate Mechanics. There are two papers here in 1994 which describe that method. And we're based in California, so I apologize to those people around the world today who are waking up early or late at night. It's 9 o'clock here in San Diego. So today's topics are how to screen or dock a database of chemical fragments to a protein target, then how to analyze and view those results, and how to sort and score and look at the scores of those and the p-values. Then how to cluster and select fragments based on their location in the ligand binding pocket, so you can dock and then you can filter based on the contacts between your fragment and the protein. Also embedded in the software is a way to envision if you have a lead fragment, and you can also view how that fragment binds in similar protein structures from the PDB. So it's a useful way of evaluating how good your docking pose is for your fragment. And then we will end with a couple of different scenarios whereby you may have a crystal structure with your fragment bound, or maybe a model, and then how to grow that fragment to make it more drug-like, bigger, and then how to, if you have multiple fragments in a pocket, how to then link those using a fragment linkers that we have in the 3D Ligand Editor. So just a sort of introduction to fragment-based drug discovery. So it's a method to identify small chemical fragments which might bind only weakly to your target. The fragment is then grown or combined with other fragments to produce a higher affinity lead. Generally, when people are talking about fragments, they sort of referring to chemicals that have molecular weight less than 300, log P less than 3, a number of hydrogen bond donors and acceptors less than 3, and number of rotatable bonds less than 3 as well. So it's kind of like a Lipinski's rules, but for fragments. The advantage of fragment-based drug design over high-throughput screening is that you're likely to find more hydrophilic hits, where hydrogen bonding is more likely to contribute to affinity. You also start off at a better position in terms of ligand efficiency, because you're starting off with a smaller lead compound. And fragment screening, fragments may find cavities which would be probably sterically blocked if you were screening much larger, more drug-like chemicals. So you can fit these into smaller cavities in your pocket, which might not be found if you're screening larger drug-like databases. And just a couple of papers. There's a famous one, JMEG Chem 2004, which describes fragment-based drug discovery. And there's also a very good blog here called Practical Fragments, if you want to read more about it. So in terms of ICM, in MolSoft, so in ICM we provide a quantitative, one way of, we have a database of chemicals fragments from the PDB, from the Protein Data Bank. And it provides a quantitative measure of confidence that a fragment is a true active and binds in a predicted specific pose. So for each fragment we have in our database, we have examples of this fragment bound. So, so it's a useful way of, it's a way of being able to validate the scoring of the chemical when you dock it. So the fragment data, this fragment database that we have inbuilt into ICM is relatively small. It's 120 fragments, but we have additional larger databases from Enamine and Maybridge that you can screen as well. But the advantage of this 120 fragments, which we'll show first of all from the PDB, is that it's relatively small, and it's been possible to we've done pre-validation of the scoring for each member of this library of 120 fragments. And this allows us to estimate sensitivity and specificity, so true positives and true negative rates at any given docking score. So this kind of results in gives you an expected number of false positive binding poses below a certain cutoff, and it provides a quantitative measure of confidence that a fragment is a true active and binds in a specific pose. So rather than just using a docking score, we also have like statistics based on these poses of these fragments based on atomic structures from the PDB. So practically, how do we do this in ICM? We would you need to load the load ICM, click on the icon, and you get the graphical user display here. I'm going to start by showing you that small database that we've extracted from the PDB of fragments. It's called, if you go to File, Open, and go to your ICM distribution, which will be in Windows, it's in C drive, Program Files, and then MolSoft. MolSoft LLC here. It's a file called it's called fragments.icf. Fragments.icf. So these are all the fragments from the PDB. So you see we have the fragment here in the mol column. You scroll down it, and we have additional scoring that we use when you dock them, and then you have also a column with the example PDB, so you can go to these PDB files and see this fragment bound. So it'd be interesting just to look at the properties of these fragments that we have in this smaller database. So we can look at those. You can go to the chemistry menu and you can calculate properties. So we can calculate chemistry, calculate properties. You can choose molecular weight, log P. We can choose number of hydrogen bond acceptors, number of hydrogen bond donors, number of rotatable bonds. So those are the key properties that describe a fragment. So we just go OK, and then we can see that we have additional columns in our table just to look at these properties. For screening and docking purposes, you don't need to do this, but it's just sometimes interesting to look at these properties. So for example, we can see the distribution of molecular weight in our small database. We can select that column, right click, and choose column histogram. We can see that if we expand this plot out a little bit, so the fragments that we extracted from the PDB are from around 80 molecular weight through to around 175. This distribution. We could make a plot maybe of on any table. There's a plotting tool here, and then you have 3 tabs: plots, histograms, and heat maps. But we're going to make a plot of molecular weight versus log P. You can see the distribution here. So we do have some chemicals with some fragments in that database with log p greater than 3, but it's not exactly. It's not. These are not hard and fast rules, of course. So we can also color this plot. If we right-click on the plot, we could color the plot by number of hydrogen bond donors. So red is 1, none. Yellow is 1, and orange is 1, yellow is 2. And we can make the size of the points related to number of rotatable bonds, for example. Okay. So if you wanted to pre-filter the database, or just this is a useful tool. If you had a larger database, which I'll show you how to download later, you can use these tools to pre-filter your screening database. So you can make selections. So if you did want to remove the ones high log p, you can just select them here, and that selection is propagated to the table, and then you can delete them. Okay, so this is just a way to introduce the database that we have from the PDB. So how do we dock this? We have an example. It's a CAMP dependent protein kinase, and we need to go to the PDB. I was going to delete this table, we don't need it. It's going to go to the PDB search, and we can search for PDB structure with the PDB code 1REK. So go to search tab, PDB search. In this search tab we have other databases, but the one we're using is PDB. Just go OK. And just for for display purposes, I'm just going to color the ribbon from N to C so we can clearly see where the N terminal is. So this is a kinase, so we have the N terminal here, C terminal, and the N and C terminal are linked by this hinge region in the middle, near the ATP binding pocket. So what we're going to do is we're going to empty this pocket by deleting the ligand, and then we're going to dock those fragments to this binding pocket. So to do that, so to empty the pocket, we're just going to, we discussed this in previous webinars, but there may be water molecules that you need to keep. One way to do that is more subjective, but in this case I'm going to assume we're just going to delete those along with the ligand. So we have an empty binding pocket. Okay, so now we need to convert. So this is a PDB structure, X-ray structure. XR stands for X-ray, 2.3 angstroms. It doesn't have any hydrogens, and so we need to build a full atom structure in order to do ligand docking. So to do that, we need to convert this into an ICM object. If you right-click on the structure here in the ICM workspace and choose Convert PDB. So this will, we can, if we were doing this properly, I'm not going to do it in the webinar because it takes a little bit longer, but you can optimize the hydrogens for the optimal hydrogen bonding network in the protein. We'll keep this checked, it's relatively quick. It's going to optimize asparaginins and glutamine residues where the orientation of those residues is not clear from the crystal structure, so it will optimize the hydrogen bonding network for these residues as well as histidines as well. I'm just going to choose the option replace the original, so it overwrites the X-ray structure. So just go OK, and now it's converting that structure to an ICM object to be docked. Which takes a minute, less than a minute, hopefully. Yeah, now it's converted. So now you see it no longer says XR here, it says ICM, so it's an ICM object. All the hydrogens have been added. So we need some way of defining where we're going to dock our fragments to. So we can use the ICM pocket finder to do that. We can right-click, double-click here to select the structure, and then we go to Tools, 3D predict ICM pocket finder, and you can, these tolerance level is sort of designed to find drug-like pockets, so you can just go OK, and it will find the ATP binding site along with other, so this the blue is the ATP binding site. There's a number of other allosteric sites on this kinase as well, little pockets that could be targeted. But the one we're interested in this example, we're going to target the ATP binding site, particularly in the hinge region here. Okay, so I'm going to delete this. We don't need that. Okay, so color my into C again, just for clarity. So now we're ready to set up the docking for the fragments. So we go to Docking menu, go to New Project, give the docking project a name. So it needs to be more than 4 characters, like this. Then you need to define a folder where it will save all the docking project files, so your docking results plus all the docking setup will be saved here, and all the files would have the prefix frag.doc1 before it, so we'll save it there. And then we define the binding site. You can run Pocket Finder again if you wanted to, but we've already done it. And so the pocket is G_pocket. It's called G_pocket1rec1. You can use the drop-down list here to find it. You say define site around mesh, and that the mesh being the blue structure. So this is where we're going to build the maps. So we go OK, and as we discussed in previous webinars, we have this probe here, and so in fragment docking it may be useful to move it to a region that you're most interested in. So basically you can use the middle mouse to move the probe, and wherever the probe is initially, that's where the docking will start. But the docking the method we have is, which we discussed before, is a stochastic random sampling. So it will start here, and then the ligand will randomly in a flexible way dock around the pocket. But you can start your search maybe in the place you're most interested in if you wanted to, or you could just leave it as default and press go. So in fragment-based drug design, this box, you need to be careful because we're targeting this cavity here, right? So if the box is too big, these fragments are going to fit in these cavities, smaller cavities around the outside of this pocket. So if you want to really focus your docking, you may want to make this box smaller and sort of to hug the region that you're most interested in docking to. If chemicals do dock, sort of peripherally to the binding site, we do have a way of filtering those out, which I'll show. But it's pretty useful to make a nice to make the box a little bit smaller to encompass the binding pocket. In regular docking and with larger drug-like ligands, they wouldn't fit in this region. So, but I think maybe some fragments might. So we just do that. You can use the left mouse to drag this purple box and change the size of it. Once you're happy with the size, you just go. This will generate the energy maps for docking. So it takes a little bit of time, a minute or so to build those maps. But once they're built, they're ready. Your docking is already set up, and you can reuse those maps again. So you could actually start from a clear ICM session, edit, delete all, and then go docking, and then open project and give that docking project name frag.doc1, and it will load everything, and you'll be okay to start docking again. Okay, so now the docking project is set up. If we go to our that directory, you can see in the directory, my docking directory, I have these files, map files, a log, the receptor object, and the .dtp, which is a table file which you can edit when you can either edit in the GUI, going to GUI preferences, and there's certain preferences to change, but the default for this example is fine. And you can see now it's set up, we also have an additional object in the ICM workspace called fragdoc1_rec. So this is the object we're going to actually dock to now. So you have the original file, and now we have this receptor here shown in red. So to dock that small database of 120 fragments, you can go to docking, you go to scan probes fragments, and then you get this dialog window. It asks you for the docking effort. This is the length of the docking simulation. If you want to be extremely thorough, you could increase it to maybe 10. If you have a very large database, you may want to reduce it to 1 just for speed. But a value of 3 is reasonable for this, for fragment docking. Then it's going to ask you how many poses do you want to score for each ligand. So it's going to dock that fragment and then take the 30 best energetically favorable ligands, fragments, and then score those. So 30 is the value for him. And then, as I mentioned, in this database we have some statistics. So we have p-values and we have percentiles. And so you can just remove those with a percentile greater than 50, and that will won't report those results. And that database that I just showed, the fragments.icf file, you don't need to select it. If you just keep default, it will dock that file automatically. And the license type for people that are using the license that came with the webinar, just leave this blank. There's some other different licenses for clusters shown here, but for your example, we just go okay. At this point, I would suggest, I sent an email this morning to download the C version. We found a small problem with the B version, so you need to download 387C, and it will dock your chemicals. So you go OK, and that runs. Okay. So when the docking is running, you'll see a background. It will say background job at the top of the graphical user interface. You'll see a progress bar at the bottom. And when it finishes, it will automatically make you a hit list. And I'll show you this hit list. So in the hit list, we have this p-value column and some other columns which I'll show now. So the docking would be running. It's pretty fast for the 120, but it's not really practical just to show that in the webinar. So I have the results file already prepared. So when the docking is finished, you'll get a little dialog box saying make hit list. If you don't see that, you can go to docking and then make hit list yourself, and that will generate this hit list. So you'll see this hit list at the bottom of the screen, which is a chemical spreadsheet. So in regular virtual screening in drug-like compounds, you just have the score column. So generally you sort by score, and the lower the score, the better the predicted binding. But as I mentioned, we have some other predictive measures of confidence that a fragment is a true active based on the PDB evaluations that the way that this fragment binds in the PDB. So we have the p-value. So generally for this data set, it's good to order it by p-value. So you just click on the column here, and it will sort by p-value. So we can see if we wanted to look at the fragment with the lowest p-value, you can just do a single click here, and that loads this fragment into the object here. So you can see now we have the protein chain and this M, which is the fragment. If we want to double click and zoom in, we just go here. And you can see this fragment is making two hydrogen bonds with the hinge region of this CAMP kinase. It's going to display a little bit differently. If you click and hold on this button, you get this option called Elegant Ribbon Ligand Sketch. And so you can, let's just make it a little bit clearer to see. So this is the top ranking ligand. If we wanted to compare it, say, with another ligand that has good score and good p-value, so low score, low docking score, and low p-value, we could we could make a plot, for example, and plot any of these columns here. So they'll find score and p-value. P-values here, so we can plot those two. So you can see that we have some. This chemical has, I think this one actually, -41, has a low score and low p-value. We've got others with quite pretty low, very low score as well as a reasonable p-value. So these are also interesting. So if you select here, this one, you could load this one into the structure, and this one's actually out of the pocket. It's away from the hinge. It's in another part of the pocket. We'll discuss that later on, how we can cluster those. And same with this one. So for us, this one is the best scoring one for p-values. You can compare with other ligands. I think this one is also in the pocket. So you can toggle the display of other fragments just by using this L button. This toggles them on and off, so you can compare the binding pose with other fragments. And the hydrogen bonds are represented by these spheres. The green is a good energy hydrogen bond between the ligand and the receptor. I'm going to untoggle that. So it's quite a big pocket that we actually dock to. So we're going to find fragments in different regions of the pocket. So it's useful to be able to cluster the fragments by where they bind in the binding pocket. So we have a column called location, and then we can cluster this column, and you get a cluster tree. So basically all the ones with similar color, we're going to be in the same, roughly in the same position in the binding pocket. So I'll show you how to do that. So we have a hit list, and we have as I go through the columns. So this is the score. This is the docking score. Generally, we assume that under -32 is a good score, but that's not generally the case for fragments. -32 is more for drug-like chemicals. But as I mentioned, in fragments, p-value is important. N-atom is the number of atoms. N-flex is the number of rotatable bonds. H-bond is the hydrogen bonding energy, so lower the better. Hydrophobic HPHB is the hydrophobic energy in exposing a surface to water. This is Van der Waals interaction energy. This is the internal conformation energy of the fragment. You're not going to see great high energies here because they're not particularly strained because they're small molecules. Desolvation of exposed hydrogen bonds and acceptors. This is the solvation energy. MF score is called the potential of mean force score, which is another commonly used scoring function in virtual screening. So you can compare that with this score is the ICM score, this is the PMF score. You can compare the two. So p-value and percentiles. So as I mentioned, because the fragment database is relatively small, we've been able to validate the scoring individually for each member of the library. So this allows us to estimate sensitivity and specificity, and provides a quantitative measure of confidence that a fragment is a true active and binds in the specific pose. So, and if we go along further, we see this column location. So to cluster by location, we can select that column, single click on the column header, and then we have a clustering button on the table here, and that will run the clustering. And you can just leave everything as by default. If you select the column, it's going to select, it's going to cluster by this column called location. I think if you by default it will cluster by the mole column, but you need to select that column and go OK. And then you have this tree. So you can see the yellow, the coloring is propagated here into the table, and we have different clusters. So if we click on this cluster, this will take us to a different part of the pocket, probably outside. Let me just check where it is. Yeah, that's outside. So all these yellow ones are in the hinge region. You can see here. So if we go through these, they're all clustered in the same. These fragments are all clustered in the same position. You can change the distance of this clustering, so then you can maybe it will differentiate between different parts of this of the hinge, or down here, up here. So it's going to change the clustering a little bit more. So if we move on to the blue ones, it's over this way towards, these are binding towards the DFG region of the kinase, away from the ATP site. Okay. So if you just wanted to, if you're just interested in the ones that are in the hinge region here, as we are in this example, we can just select. You can use the shift and select, and that will select all of the ones in this region. And then you can use the tools here to delete the others that you're not interested in. Or you can select. You can right-click on the table on the clustering tree here and choose select a representative center from each of the branches of your tree. You could say select by minimum p-value here, and it will select a representative from each branch of your tree, and you can make a smaller hit list here. Okay, so this was our best scoring one in terms of p-value. So you single click and it shows that ligand. So it'd be nice to see how this fragment also binds in other structures from the PDB, so we can compare it. So each fragment in the library is linked back to the PDB entry from which it came from, and and it also has the appropriate rotation translation matrices, and it instantly superimposes it from the PDB that the fragment was taken from. So I'll show you how to do that. You can see another column here called example PDB. If we click here, it's loading, I think, by default. Some fragments have multiple examples. So for this example, this fragment is seen in 52 other PDBs, but by default it's going to load the first 10 and stop. So it loaded the first 10, and so we can see that this the fragment. Let me just. Okay, so our fragment is here, and you can see the other ligands from other PDBs bound. So if we say color by, it's got undisplay here. We can say color by object C, and the ligands all from other PDBs are displayed. So you can see the fragment is from, it has the PDB code 3NUP for one of them, 3UNJ, 3UNZ, and they're all automatically read in and superimposed on your fragment. So you can see these are all kinase binders. It'll give you ideas as well how to, if we just not display all 10, just display some other ones, it'll give us ideas on how to grow this this fragment. If you want to sort of copy other inhibitors from other proteins using this fragment, it gives you an idea where maybe you can grow, and also some ideas for the chemistry as well. So that's another way of evaluating the results. So this is, you'd screen this, you can screen this small database relatively quickly, and it will give you some ideas of useful fragments. So we also have additional fragment databases, particularly we have two, one from Enamine and one from Maybridge. If you go to Help, go to the Help menu in the GUI and go to MolSoft add-ons, you can download the database. And you dock them in the same way. You set up the docking project as we did earlier, and then you go to Docking Scan Probes, and you enter the full path for your database that you downloaded, so for example the Enamine. But you dock them and you make a hit list. And because these fragments aren't extracted from the PDB, we don't have the p-values, percentiles and stuff like that for these fragments. But what we do have is a z-score, and you should choose the ligands that have a low docking score and a z-score. So the z-score helps to filter out those fragments that are getting excessively high docking scores because maybe they make a lot of hydrogen bonds or salt bridges. And so it's a useful scoring. So you're not just using the docking score, which may be biased by a fragment that's making 3-4 hydrogen bonds with the receptor, and this will filter those out. So you can compare the z-score with the docking score, and then choose those that have low docking score and low z-score. So I just want to show you practically in ICM how you do that. You would go to Help, go to MolSoft Add-ons, then you will see a web page like this, and there are a number of different options. One of them is called Screening Fragments here. And then you can just download the SDF files from here. And then you would go to Docking. I already have set up this project, as you remember. So you go to Docking, Scan Fragments, and then just enter the path to that SDF file here and click OK, and it will dock those. Okay, so I have some questions, but I'll get back to those at the end, I think. So that's docking and screening. So we're now moving on to a scenario whereby maybe we got lucky, we found a fragment that binds experimentally in our binding assays, and we've also we also have a crystal structure of the fragment bound. So just going to so imagine we had a PDB, we have the receptor structure shown here. It's another kinase example. And we have a small fragment bound. How can we grow this fragment and design it? So the first webinar in this series was based on the Ligand Editor. So I'm just going to quickly show you how you can use that to design and grow this ligand. So we have here we have some tabs: display, lighting, labels. The last one is called Ligand Edit, and the Ligand Edit is the one we need to use. So when you click on that tab, you see some buttons. Basically work our way across these buttons. The first one is to tell ICM which is the fragment or the ligand. So we click on Set Up Ligand, And in this example, it automatically finds the fragment because there's only one fragment in our PDB that we just read in. So we just go OK. You can assign charges if you need to, and go OK. And then we need to tell ICM which is the receptor. So we click on the button next to that called Setup Receptor, and the receptor object is called 1xbb fragment bound. It finds it automatically. It needs to convert it to an ICM object, as we did when we were using the docking menu. So you can optimize hydrogens, asparaginins, glutamines, histidines. And in this case, we don't need to run Pocket Finder because we have the ligand already bound to the protein. So we can say make box, which is the energy maps here around existing ligand. So we can once again change the size of these if we wanted to. Just drag on the corners of the box, and we go OK, and then it'll make the maps. So there are many tools inside the Ligand Editor that you can use, and a lot of those were covered in the webinar, the first webinar in the series, which you can look at in YouTube. But by default, it shows this ligand pocket surface, which gives you ideas of, which shows you where there's space to grow your fragment out. So we have space here, we have space here around this section. It's completely closed off here. If you add any substituent on this point, it's going to make a clash with the receptor. So there's different visualization tools that you can use. You can, well, the one I want just by default is going to be Pretty View, and so we can clearly see that our ligand is making one hydrogen bond with the hinge. Sorry, our fragment is making one hydrogen bond with the hinge here. We could look at the key interactions. We can display the receptor pocket. So we can see this is colored by binding property. So green is hydrophobic, red is hydrogen bond acceptor, blue hydrogen bond donor of the receptor structure. So we can undisplay that just for this case. If we want to start growing the ligand, we could maybe superimpose those structures that we had shown previously with similar fragments. That would give us some ideas of modifications we can make. Or we can click on the modification button here. We get some groups that we can try. So we can, you can just try one or more. You could just make one modification of one. You could add a chlorine, see if that makes any difference. You get a binding score here, the VLS score, which is the lower the score, the better. And in the editor, you can just undo to go back to the original pose. You can do virtual screening on the fly, so you can select multiple substituents and just click on the region where you want to try them, and it will screen those, and you get a VLS score. So the lower the VLS score, the better. So this addition here actually improved the score, probably because we forced another. This addition actually made an additional hydrogen bond here with the hinge region. So we have two hinge. If we don't like that modification, we can undo it and go back to the original one. So there are a number of different tools. You can also edit in 2D your fragment, and then apply the changes to 3D by clicking here. And you could do a substituent screen on the fly using Find Group. This will screen a larger database of substituents at certain points. And you can play around with different modifications to try and improve your binding affinity from your original fragment. But I would refer you back to that original webinar for all the different tools that you can use here. So you could actually tether your original fragment in place from your crystal structure, and then redock redock the ligand using this button here. This will fully redock this ligand but keep this original fragment in place. So there's quite a large number of tools here to do the fragment growing and improvement. So you see it's docked and we have different conformations of the ligand in a table here. If there's a modification you like, you can add it to you can save it to a spreadsheet and come back to it later just using save ligand, or you can tag it, give it a name and or a series name, and then you can always go back and you can always undo your modifications, and you can always see the history that you've done. So if you click and hold on the undo button, look at modification history, you can see all the modifications that we've tried. Not too many on this example, but if you want to go back to the original ligand, you just double click on this table and it will take you back to that point. Okay. You may also be in a situation where you have two fragments bound in your binding pocket and you want to link those fragments. So I'll just have a quick example. So this is all done in the Ligand Editor. So the Ligand Editor, which I've just shown, is we developed with Novartis, with the medicinal chemist there. So it's quite user-friendly, designed for non-computational chemists. It's designed so you can explore different chemistry quite easily with full undo and redo capabilities. You can dock, induce fit docking, which we covered before, and you can also design in 3D using 3D pharmacophores. But we also have a tool that allows you to link fragments. So you may be in this example where you have two fragments in the binding pocket and you need to find a way of linking them. So what you do is you interactively select two or more points for connection, and then inside ICM we have a database of linkers that you can screen. And we have a bond called a cutting bond, which I'll show you, which defines where you want to link, between which 2 points you want to link, and then you get a hit list of linkers that fit. So to practically do that in ICM, you would probably I'm just going to show an example. So you might be in this situation. We have a crystal structure with maybe a density here for one of your fragments, and another density here for your other fragment. You'd go to the Ligand Edit tab and you would set it up the way I just showed for the single. There's no difference. And then in order to screen linkers that can fit between these two fragments, we need to first click on this button here, and then this is the cutting tool button. So this will just show you, I want to show you, so you can click on this cutting tool and then click where you want to, has this little arrow here for one fragment, and we want to connect it to this hydrogen here. And so we'll then go to Advanced and we choose Find Linker. And you can screen a database of linkers that you've already built yourself, or you can screen our database that we have built into ICM. So you would need to actually download the linker files. If you go to the Help menu, MuleSoft add-ons, we have here we have a linker database from Cambridge, and a database of linkers that were made from drug bank. So you could choose either one of these and then browse for that file. It's called a multi file, MOLT file, and then you have certain cutoffs. So we have it's quite easy to find linkers, but the linkers may be strained. So we have an energy value for the strain of that linker, you can define maximum number of rotatable bonds that you want in the linker to be 5. There's a default value for the contact with the receptor cutoff. So any ligand that exceeds this distance will not be placed in the results table. And you can also have you could place a 3D object here and use it as a pharmacophore to guide which linker you'd prefer, what kind of properties you want in your linker. So if you you can use the drawing tools here and sketch a 3D object for maybe like a hydrogen bond donor or acceptor or something here at this point, and it will try and match when it searches the linkers between these 2 points. It will try and match the pharmacophore as well. Okay, so when it's done, you go to Advanced, you go to Find Linker, and it will search that linker database. And then you will get a hit list. It's going to show you. Something. Oh, sorry. Okay, you will see a hit list here with the link. So you can just double click, and that will load that linker between those 2 fragments. The fragment, and they're ranked by a score. So basically the score is the energy, you have the strain, so it's the energy, the strain of the ligand, and this is a score, so the lower the score, the better for these linkers. And you can toggle through them, you can sort by score, you could do a plot of score versus strain, for example. If you had a pharmacophore 3D object here, it will tell you how different your linker is from that pharmacophore in terms of RMSD. And it also tells you where they extracted the linker from. So it has the name of the molecule it took the linker from, and the drug bank ID code as well. You can also link you can also do 3-point core replacement as well. So just want to, I don't have an example, but in the manual it shows you that you have three fragments. You can also find more than one. You can find linkers between 3 as well. Okay.